Hello friends, how are you doing? And I hope you really are having a great and fun time. Things are going smooth, you are sailing in the right direction, and yes, this is the way to go. Go about things very smoothly. Slow and steady wins the race. And yes, it is the speed always thrills, but it kills. Okay, it does not apply here. Okay, jokes apart, we had done till the fifth question, the pedigree analysis. We move on, and not in I mean in terms of question I'm talking about. Move on. In pea flower, how many stamens are free and how many are fused? Why should we bother about the pea flower first of all? Okay, I know we are not bothered about the pea flower, but when it comes to entrance exam, we have to be bothered because this will fetch us a seat. Yes. First of all, when we see a question pea flower, in pea flower, how many stamens are free and how many are fused? So the moment this question comes, what you should do, leave the pen, okay, and leave the exam hall and go out to the garden and see whether you find a pea plant, so that you can see the number of stamens and you can count them. Right? This is the thing you should do. Not at all. Don't dare do that. Otherwise, okay. So my dear friends, let us see the options. The options are in the A 1 9, in the 2 B option 2 8, 5 5 and 4 6. Definitely one thing is clear from these options. If you do not know anything, one thing you will get to know from these options, that the pea flower has 10 stamens in all. Yes. 1 plus 9 10, 2 plus 8 10, 5 plus 5 10, 4 plus 6 10. So there are 10 stamens in the pea flower, and you have gained this knowledge right now. So let us write the answer. The pea flower consists of 10 stamens in all. Okay, the pea flower consists of a total of 10 stamens. Right? This we know, this is a known fact, and in these 10 stamens, the arrangement we see is 9 plus 1 arrangement. Okay? So the first option will be right. There is 9 plus 1 arrangement. There is 9 plus 1 arrangement of the stamens in pea flower, in pea plant. Right. The filament of these 9 stamens, they join through most of the length of their, of the filament. Okay? The filament of, of these 9 stamens joined almost throughout their length, that is much of their length, to form a staminal tube that will be surrounding the ovary. Okay? So the filament of 9 stamens will be joined right? The filament of 9 stamens will be joined through much of their length through much of their length to form the staminal tube. To form staminal tube, and it is this terminal tube, okay, and it is this terminal tube which will be surrounding the ovary, and it is this terminal tube which will be surrounding the ovary. Right. So, the filament of 9 stamens will be joined through much of their length to form this terminal tube, and this terminal tube will be surrounding the ovary. The 10th one, that is the 10th stamen which we will see, will be it will not be fused, it will be free. So, the 10th stamen will be free. The 10th stamen will be free. Right. Now we see one more thing. When the filaments are young, okay, when the filaments are young, what will we see? The length will be different. The length of the filament will be different. They will be shorter than the style, but on maturity, when the pollen grains are shed, they reach, the length increases, right? When young, the filaments are shorter than the style. The filaments are shorter than the style. Okay, so when young, the filaments are shorter than the style. However, they elongate by the time of pollen shedding. By the time of pollen shedding. Okay. So, we have done the explanation of this particular question. Once again, in pea flower, how many stamens? We see 10 stamens are free and how many are fused. So, the 10th stamen will be free, and the other 9 stamens, what are they doing? They are joining. They are joining to form a staminal tube. This staminal tube will be covering the ovary, and we see when in the young stage, the filament are of not of the adequate length, but at when maturity they increase their length, right? So, we have come to the end of this question. Let us see the next question about, okay, first when Mendel allowed natural selfing of the hybrids during the monohybrid cross between a pure tall and a pure dwarf pea plant, he found all plants were tall, all plants were dwarf, dwarfness reappeared in some plants, and tallness reappeared in some plants. Let's see what will be the answer of this particular question. Let us read the question again with some impetus. When Mendel allowed natural selfing of F1 hybrids, so there will be selfing of the hybrid F1 generation over here, but how will be the F1 produced? By the selfing of the, not selfing, by the crossing of the parent. So, what will be the parent? During monohybrid cross, that is involving one trait, the cross will be made between a pure tall and a pure dwarf pea plant. So, This is what? This is the parent. This is the parent, and you know, in these parents, among these parents, the crossing will be done for a monohybrid cross, and whatever F1 generation offspring we are getting, whatever F1 generation offspring we are getting, we have to make them go the natural selfing, and then we will obtain the F2 generation. And then in the F2 generation, the question is telling what can be the probability among them. Will all the plants be tall? Will all the plants be dwarf? Dwarfness reappeared in some plants and tallness reappeared in some plants. So let us do this cross. Okay, come back here and let us see what this question says. So this is question number 7. Parents are what? Pure tall and pure dwarf pea plant. Pure tall and pure dwarf pea plant. Parents are pure tall and pure dwarf pea plant. So if I write the parents, what will be they? Capital T, sorry, pure tall. So homozygous dominant condition we have to take, and the homozygous recessive condition. So this is homozygous dominant, right? And this is the homozygous recessive condition, what we will see here, right? This is the homozygous recessive condition. Now what will we see? We will be crossing between the two, okay? So if we produce a cross between the two, what will we obtain? Capital T small t, right? Capital T small t, capital T small t, capital T small t, and capital T small t. So a heterozygous condition, but all will be dominant, all will be tall in the F1 generation. This is the F1 generation, right? Homozygous tall, homozygous recessive, then we will see the gamete and we will cross them. Now come back to this question again. Okay, what will we see here? What will we see here? When Mendel allowed natural selfing of the F1 hybrids during the monohybrid cross. So what are we doing here? In the F1 generation, we are selfing the F1 generation, and we know capital T small t is the genotype of the F1 generation. Okay. So F2 generation is what? F2 generation is selfing of the F1 generation, is selfing of F1 generation, right? And what had we obtained? Capital T and small t. So if we self this one again, what will we obtain? What will we get here? Capital T, again a homozygous dominant condition. Capital T small t, that will be a heterozygous dominant condition. Capital T small t, a heterozygous dominant condition, and small t, right? Small t, that is dwarfness has appeared again back here. You saw that in the F1 generation, all the 4 plants, you remember? Okay, first I'll write this F2 generation. This is F2 generation, right? Now what are we seeing here? In the F2 generation, we will see these things. In the F1 generation, when we talked about, what did we say? In the F1 generation, we obtain all 4 tall plants, right? All were of the genotype capital T small t. And however, in the F2, how many tall plants we are obtaining? There is 3 tall plant and 1 dwarf plant. 3 tall plant and 1 dwarf plant. Three tall plant and 1 dwarf plant. So we see that this dwarfness character has reappeared back in this F2 generation, and come to the options again. Okay, we now know the results. All plants were tall. Is it in the F2 generation? No. 3 were tall, 1 was small. All plants were dwarf. False. Dwarfness reappeared in some plants. Yes, it reappeared in the ratio of 1 out of 4. 1 was dwarf and 3 was tall. Tallness reappeared in some plants. So wrong. The answer will be C for this particular question. The answer will be C for this particular question. Right. Let us go to the next question now. And what will we see in the next question? The law of dominance is illustrated in the garden P by the heterozygous tall into heterozygous tall. The homozygous tall and the homozygous tall, the pure short and the pure dwarf, and the homozygous tall and the pure dwarf. Right. What we did just now for the dwarf, for the tallness in P plan, is absolutely the same thing what is asked in this question. See, according to Mendel, according to when Mendel did the first law of dominance, the first law of Mendelian inheritance was called as the law of the dominance. And then we had the law of the segregation, okay, and totally there were 3 laws given. The law of the segregation. So what we see here, Mendel, he took pure tall P plants and pure dwarf P plants, that is capital T and small t. He took pure plants of a monohybrid cross of tallness. He, crossed them. He crossed them, and what he obtained was, all the plants, all the P plants in the F1 generation were absolutely, tall. None was dwarf. 4:0. Then he self-crossed them, and then he obtained plants in the ratio of 3:1. That is, 3 were tall and 1 was dwarf. So that is what I said. We are doing the same thing again. Okay, the dwarf plants reappeared back, and hence the law of dominance in the is illustrated in the garden P by homozygous tall into pure dwarf. Let us see what the law of dominance says, right? I'll just write them down. Mendelian laws of inheritance. We have the law of dominance. after the law of dominance, we have the law of segregation, and then we have the law of independent assortment. Right. So these are the three laws which were given by, in the Mendelian's law of inheritance. The law of the dominance, the law of segregation, and the law of independent assortment. Now, basically what we consider is the law of dominance as the first law, and but sometimes, many of times, in some of the books they have not given so. But for our reference, we will consider the first law as the law of dominance and the second law as the law of segregation. Okay? Many in some of the books tells different things and different stories. Forget about that. Now we will talk about the law of dominance, which we have considered and is considered in most of the books as the first law of Mendelian inheritance. What does the law of dominance says? The law of dominance says that every character, every trait in an individual or in an organism is controlled by a pair of factors. Out of these pair of factors, one is dominant and will be expressing the phenotype, and the other will be recessive, which is not expressing the phenotype. That recessive allele will be expressing its phenotype only in the presence of another similar allele. Okay? So, law of dominance, what does it say? States that number one, every character is controlled, every character is controlled by paired, or rather than paired we can write discrete, by discrete units called factors. These factors occurs in pairs. These factors occurs in pairs. In dissimilar factors, or when the factors are dissimilar to each other, one will be dominant, one will be dominating the other, and it's called the dominant type, and the one which is being masked is called as the recessive type. I'll write the third one there. I hope is it senior? Is senior? So, what have we written? These factors occurs in pairs. When these factors are dissimilar, when these factors are dissimilar, one will be expressing itself, called dominant factor, called dominant factor, and other called as recessive factor. Got it? So when these factors are dissimilar, one will be expressing itself and called the dominant factor, and the other will be called as a recessive factor. This is a very, very important thing to be noted. And that is what the law of dominance said. That the law of dominance is saying that, okay, every character is being controlled by two pairs, two unit factors. Okay, by pair of unit factors, one will be dominant, other will be allele, and the one which will be expressing itself in the absence or in the presence, we can say, of dissimilar allele will be the dominant one, and the other one will be the recessive one, and this is the law of dominance. So what was done? A homozygous stall condition, that is a homozygous stall condition into homozygous, okay, pure dwarf condition was taken, and then we, it was further, the crosses were made as to what we, did in the last, question, right? The law of dominance. So the answer would be the D, that is the homozygous stall into pure dwarf. That is why we, okay, if you cross this, what do we see? T, we'll get capital T, small t, capital T, small t, capital T, small t, capital T, small t. And if we take this capital T, small t over here and again self them, what we see? Capital T, capital T, capital T, small t, capital T, small t, small t. That is exactly what we did in the last session. I mean last class. No, sorry, the last question I'm talking about, not the last class or last session. So we see here that in these cases, this is a heterozygous condition. Both the gametes are separate. Capital T, small t, capital T, small t, capital T, small t, capital T, small t. And even the presence of these dissimilar gametes, what we are seeing in the presence of this dissimilar gamete, we are seeing that one will be expressing, that is capital T, all these are tall. All these four are tall. One is expressing, the other is getting masked. This is a recessive, this is dominant, recessive, dominant, recessive, dominant. Here also what do we see? Here also what do we see? That all these are tall, but this is small t, small t. Here this will become the small. Okay, so with this we have completed this question. Two more questions remaining. We'll take a short break and we will be back soon after a small time. Okay friends, so I hope you must have noted down. Now let us talk about the next question, the ninth question. Reappearance of recessive trait in the F2 generation is due to law of independent assortment, the law of dominance, the law of codominance, and the law of the purity of the gametes. Right. Once again the same thing. When we talked about the tall, the crossing between the pure tall plant, that is homozygous dominant, capital TT, okay, capital TT, that is pure tall plant, into small tt, pure dwarf. Right? We obtained capital TT 4 times, capital TT, and capital TT, and on selfing what we obtained was capital T is to small t. What we obtained was capital T, small t, small t, capital T, capital T, capital T, and small t, small t. The ratio of genotype of 1:2:1 and a phenotypic ratio of 3:1. So here we see this was the parent, right? This was the parent group. This is the F1 generation, and this is what? Sorry. This is the parent group. This is the F1 generation, and this is the F2 generation. Okay? So, right? This is the F2 generation. In the parent generation, we see that there is a pure dwarf plant. In the F1 generation, there is absolutely no dwarf all at all, because capital D, the dominant allele, is present in all. But however, in the F2 generation, we see that there is one small t, and hence there is one dwarf plant. So, reappearance of recessive trait in the F2 generation. You see that in the F2, this recessive trait, why recessive trait? Because it is expressing only in the presence of two recessive alleles, so it has become dwarf. It has come back in the F2 generation. In the F1 generation, it was not there. This is due to, I will say, the law of independent assortment. Sorry, the law of the purity of the gametes. The law of the purity of gametes. We will see what the law of purity of gametes mean. Okay, we know that the first law of Mendel is called the law of dominance. Law of dominance. The second law is called the law of segregation. The law of segregation, or the law of purity of gametes, or law of purity of gametes. And the third law is called the law of independent assortment. Okay? The law of independent assortment. Right. Now, what is the law of segregation or the law of purity of gametes? We will just see in one or two lines. Law of purity of gametes states that when unit factors are brought together at the time of fusion or fertilization of the gamete, okay, that law of purity of gametes states that unit factors, that when unit factors, okay, that when unit factors are brought together during fertilization, these unit factors do not mix or blend with each other, and each carries its own significance and its own trait, and this is called as the law of the purity of gamete. Are brought together during fertilization, these do not mix or blend even though they are paired. Okay? So, what do you mean? I mean by this that, okay, fine, if there is a capital T coming from one of the male gametes, say, and a small t coming from the female gamete, when they come together, it doesn't mean that small t is losing its significance. Okay, it is only becoming capital, it is becoming tall, so you know this is losing its significance in the second generation. When this small t is combining with another small t, it is going to be showing the dwarf condition or the dwarf state, and that is what the law of the purity of gametes or the law of segregation says, that even though they are segregating and coming back on fusion, okay, what is happening during segregation and during the time of cell division? They are segregating, the gametes are, you know, these are segregating and going into different gametes, the genes or the paired factors or the alleles, what you can say, and then when they are coming back, they are not losing their own significance. What they have to do, they will do, and that is why the recessive trait is coming back in the F2 generation. I hope you have understood this. What about the next question? Which of the Mendels law will always prove to be universally true in all cases? All the three laws. The first law we know is the law of the dominance, the second is the law of segregation or the law of the purity of the gametes, you can say, and the third is the law of the independent assortment. We all know that the law, the second law, the law of segregation is universally acceptable. Only the second law. Why? Because the first law we will see, the law of dominance, is not applicable everywhere. Is not applicable everywhere. In the law of dominance we see, you know, we were obtaining a phenotypic ratio of 3:1 in the F1 generation. Okay, remember F1 for a monohybrid cross, and for the double hybrid cross also in the F2 generation. So, however, the law of dominance, you know, is not applicable everywhere. Not applicable everywhere. However, second law, that is law of segregation, is universally applicable, is universally true or applicable, whatever you want to say, is universally true. Okay? Right, friends, so we have come to the end of this 10 questions, 5 we did in this session and 5 in the previous session. I hope you are following things. I hope genetics is clear, because we have discussed so many things. We have discussed the pedigree analysis. We have discussed the various rules of pedigree analysis. We have discussed all these, the Mendels Law and all those things. And as I keep on telling you, genetics is 10% of your total bio, and bio is 50% of your complete NEET. So if you have command on bio, if you have command on genetics, things will be easy. You will have that extra edge. You will have that extra, the extra thing, and definitely your chances to clear NEET would be that much high. Go back, revise this. Any doubts, you can always approach me. And the time we meet again, a very goodbye from Dr. Ashmir. Thank you.